Okay, so we're going to talk about protein synthesis here. And what we're going to do is we're going to start off with what we call the coding strand. So we're just going to put C, A, T, G, C, A. This is completely random, by the way. T, C, G. I'm just thinking off the top of my head. So again, this is the coding strand. So I'm going to label that right here. Just going to put COD to represent coding strand, COD. And then we have to have what we call a complementary strand to this. And the complementary strand we're going to produce is called the template strand. Okay? And we'll explain what the template strand means in a few minutes and coding strand. We first just have to write it down. So we know C binds with G, A binds with T, T binds with A, G with C, C with G, A with T, T with A, C with G, G with C. Now, notice that we have T's in here. So we have a T here, and we have a T here, and a T here. You're only going to have T in DNA. So you're only going to have thymine in DNA. If you had the letter U, you would know that it's RNA. Now, when we're looking at DNA, we want to remember that we read things in threes. So again, we read this in threes. So what we do is we take 1, 2, and 3, and that's what we call a triplet code. Triplet. Remember, triplet means 3. So 1, 2, 3, triplet code. Next, 1, 2, 3, and this, again, is your triplet code. So I'm going to write TC, and the next one is also going to be from here to here, so this is going to also be a triplet code. Multiple triplet codes form what we call a gene. So multiple triplet codes form a gene. So this gene could be for anything. It could be for, if it was us, it would be for, let's say, eye color or hair color or anything, really. It doesn't matter what the gene is. It's just specific, okay? Now, obviously, I'm drawing on a whiteboard, so this is significantly smaller than what you would actually see a gene be, but we're just doing this to make your lives easier and our lives easier so we better understand what's going on. Now, this right here we call the template strand, and this right here we call the coding strand, and we say that they are complementary to each other. So if you said that you had a husband and a wife and they got along well, we would say that they are complementary to each other. Okay, they match well. In any case, I'm going to put a little dotted line here, or dashes, to represent that this right here is going to be the nuclear envelope. So here we have the nuclear envelope. And as you remember, the nuclear envelope is what's going to keep the DNA inside and kind of everything else outside. These little holes right here are called nuclear pores. Nuclear pores. Okay, so the holes are called nuclear pores and the membrane is called the nuclear envelope. Now, if you get a question on an exam that asks something like, is the nucleus an organelle, your answer is yes, it's an organelle, okay? It performs a function. And if you remember, an organelle is something that performs a function in the cell. You may also get another question about, does DNA have the ability to leave the nucleus? And the answer is no. Now remember, we're talking about our cells here, so that's really an important distinction. Is remember, if they're talking about our cells, the answer is no, okay? So this DNA is never going to go outside, never. Never, ever. In any case, what we do is there has to be some way to go to right out here, outside of the nucleus, called the ribosome. Now remember, the function of what we're doing right now is protein synthesis. That's what we're doing. And this whole thing right here is called the ribosome. So keep in mind, keep saying to yourself, we're trying to perform protein synthesis. All of it starts with the gene. So let me ask some questions. Number one, must there be a complement between the coding strand and template strand? And the answer is yes. Next question, how do we read things? We read things in threes. We call those threes triplet codes. So what is this? Triplet code. What are these three? Triplet code. What is this three? Triplet code. Multiple triplet codes form a gene. Okay? And remember, the overall goal of what we're talking about is protein synthesis. We're trying to form proteins. Okay, next. So what we have is we have this little enzyme. here. Okay, so we've got this little enzyme here. And the name of that enzyme, I'm just putting a label here. The name of that enzyme is called RNA polymerase. So it's not RNA polymerase, it's RNA polymerase. And what we do is what RNA polymerase's job to do is to basically find a match, a temporary match for the template strand. Okay, so this is the template strand. The job of RNA polymerase is basically to find a temporary date for the template strand, each one of these nucleotides or nitrogenous bases, which I'll have for you in another video. In any case, RNA polymerase says, oh, okay, I have the letter G here. Who matches with G? And we all kind of know that G matches with C. So it says, okay, the RNA polymerase says, okay, I did my job. What I'm going to do is I'm going to move to the next one. So it moves to the next one, and it goes to T. And what it does is it says, okay, let me find an RNA that matches T. And the RNA that matches T is the letter A. Now remember, can you have the letter A in RNA? Yes, you can, but you can't have the letter T. Okay, so just keep that in mind. So after T is bound with A, and who marries them together, or who puts them together on a date? The RNA polymerase. After it does that, it then moves to the next nucleotide, and it goes to A. A binds with U. Now, why does it bind with U? Because this new strand that we're producing is mRNA, which stands for messenger RNA. So again, mRNA stands for messenger RNA. Okay, so let's review this. Can DNA have U in it? And the answer is no. It can only have the letter T. But can RNA have the letter U in it? And the answer is yes. So this right here, the letter U, if you ever see something with the letter U, it must be RNA. So if you see the letter U, it must be what? RNA. If you see the letter T, it must be DNA. So these two strands here are DNA, and we're creating a new strand of RNA. What type of RNA? Messenger RNA. So after it moves down, and I'm just not really going to bother with the rest of it because it kind of does the same thing. C binds with G, G binds with C, T binds with A binds with U, because remember, we see the U, we know it must be RNA. G binds with C, and C binds with G. This whole strand now, this whole entire strand that we see right here is messenger RNA. This whole strand is messenger RNA. Now, a question before I asked is I said, can DNA leave the nucleus? And the answer is no. But can messenger, the answer is yes. Can messenger? Yes. Move? Yes. From the nucleus? Yes. To the ribosome? And the answer is yes, it can move. So what it's going to do is it's going to go doot, 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 right through the nuclear pore, and it's going to go right onto the ribosome. So here I'm just going to rewrite ribosome here. Okay, so what happens, you have C, A, U, G, C, A, U, C, G. Okay, so again, what is this strand right here? It's called what? Messenger RNA. It went from the nucleus, went from the template strand to the ribosome. One thing to take note, when you're going from the template strand of DNA to messenger RNA, so when you're going from this to this, that's called translation. Okay, that's called translation. And I'll come up with another video that'll show you that as well. Okay. Okay, so this is messenger RNA. Messenger RNA right here is located on the ribosome. So if you get asked a question on a test, where does RNA synthesis take place? What organelle? Your answer is ribosome. Okay? So the way we look at ribosomes is, again, we read them in threes. Okay? So in DNA, we read it in threes. In messenger RNA, we read it in threes. So the three we have right here is called a codon. So CAU is a codon. GCA, which is the next one, is also a codon. And UCG is also a, guess what? Codon. So how many codons do we have? 1, 2, 3. And codons you only find in messenger RNA. So is messenger RNA going to be made of triplet codes or of codons? And the answer is codons. Next, we have something called tRNA. So I'm going to use a different color here because this can just be more exciting with a different color. Yay, red. Now, this is a slightly different description than the way the books have it, just in so much as the picture's a little bit different. But I'm doing it because to me, it makes it a little bit easier to understand at the beginning. If you end up taking a higher level course in biology or genetics or something like that, it'll just be flipped upside down. But I like to say that these look like ghosts or like a cactus, which a lot of my students seem to like that, is that they just remember that this looks like a cactus. One other thing to note is that it looks like what letter? The letter T. So being that it looks like the letter T, we call this tRNA, which stands for transfer RNA. So tRNA stands for transfer RNA. Now in reality, being that this is RNA, do you expect there to be the letter T, or do you expect there to be the letter U? Again, this is tRNA. Do you expect this to be the letter T or the letter A on this? The letter U. Excuse me, the letter T or the letter U, and the answer is the letter U. So what you'll have is you'll have something like this, like U, A, G, C, A, U, C, G, A, C, G, U, A, C. And it just keeps going around following that ghost or cactus-like fashion. But the most important three, because remember, we're basically reading these in three, most important of the three is the three at the very bottom. And the three at the very bottom, in this case, I'm going to label them as G, U, and A. Now, the reason I label them as G, U, and A, and keep in mind, I've been doing this for a while, so I kind of am stepping a little bit ahead of you because I know what has to come next. I wouldn't expect you to right off the bat know why I'm doing this. But the reason I put that is because this right here, these three are called an anticodon. So these three are called the anticodon. Okay, the anticodon. Now, if you remember, the 3 here on messenger RNA is the codon. The 3 on here is called the anticodon. So this is the anticodon, this is the codon. So do the codon and anticodon match up? The answer is yes. What does G bind with? C. What does U bind with? A. And what does A bind with? U. So these 3 bind together. So G binds with C, U binds with A binds with U. Perfect. Attached to each tRNA is going to be an amino acid. So I'm going to call this amino acid X. It doesn't really matter. Okay? Then what we do is we look then at this one. So we can see that we have, I know it's getting a little hard to see or observe. This right here is GCA. Okay? So what would be these 3 on the bottom of this tRNA? It would be C, G, U. Because G binds with what? C. C binds with G. A binds with U. Okay? Now there would be all the other corresponding nucleotides around there, but we don't really care about those. We just care about the anticodon. And then this is UCG. So what would the 3 here be on this tRNA? Would be A, because U binds with A, C binds with G, and G binds with C. Okay? Now, to reiterate this, these 3, because it's messenger RNA, is called a codon. These 3, which are on tRNA, that thing that looks like a T or a cactus, okay, or a ghost, is called the anticodon. Take note that anti has what letter in it that codon does not? T. So the letter T in anticodon tells you it's part of tRNA. So again, this on messenger RNA is called a codon. These 3 on tRNA is called an anticodon. So what are these 3 called if they're on messenger RNA? Codon. What are these 3 called? Anticodon. What are these 3? Codon and anticodon. Now, attached to each tRNA is a what? Amino acid. So again, attached to each tRNA is a what? Amino acid. And if you remember from your earlier courses, amino acids are the building blocks of proteins. Or another way of saying that is amino acids is what forms proteins, what makes it up. Okay, it's parts of it. So we're going to say that attached to this one is amino acid X. Attached to this one is called amino acid Y. And attached to this one is called amino acid Z. Okay? When this tRNA binds like this, so I'm going to put the black one binding like that, this amino acid jumps over to there, and they form a bond. So I'm going to erase this where it says RNA polymerase, and it's going to form a bond. So now amino acid Y will be bound to amino acid X. Now you have two amino acids linked together. We're starting to form a larger protein. Now, when this tRNA binds to this codon of messenger RNA, amino acid X and amino acid Y jump over, and now we have amino acid X bound to amino acid Y. And what are we forming? 1, 2, 3 amino acids. And amino acids are the building blocks of proteins. So in essence, are we forming a protein? And the answer is yes. So again, the goal is to form this, because this whole process is called protein synthesis. Keep an eye out for my next video, because my next video is going to be questions regarding this topic to make sure you completely understand it. And if you don't, if you get some of those questions wrong, you can always go back and review this process again. Thank you very much, and enjoy your next videos.